Okay, so thanks and good morning, everyone. I'm going to talk today about our recent work on Pierce's disease, and you can tell by everyone involved that it's been a very large project and it's gone on for about 5 years now. So, and it's been funded by the PD GWIS board, which are the grower assessments. So thank you to everyone who continues to pay their assessment and support the continued operation of the PD GWIS board. Okay. All right, so Pierce's disease, what's important about the PD system, we have vector, we have host, pathogen, of course, the environment, your sort of typical disease triangle. But what we also have are alternate hosts for both the pathogen and the vector that come into play. So I'm going to talk about all of these different components, and then we'll go into management a little bit more. So the pathogen for Pierce's disease is Xylella Fastidiosa. It's a bacterium that is limited to the xylem, meaning that it lives only in the xylem. And because of the populations that build up in the xylem, that's going to affect water transport within the vine. And those effects on water transport then affect the growth of the vine and lead to what is an incurable disease ultimately. Grapevines are not the only commercial crop that is affected by Xylella. So almond leaf scorch is also quite common. Peaches are affected, plums, citrus. And for anyone who's been following the olive situation, particularly in Europe, olive leaf scorch has emerged as a more recent issue. In order for Xylella, because it's within the xylem, to move between hosts, it requires a vector. And in that case, this vector are a suite of insects, and they feed on infected hosts, pick up the bacterium. It's in their mouth parts, which you can see here. And then as they feed on a healthy host, they're able to transmit. So the bacterium then moves out of their mouth parts and into the xylem of a healthy host. These xylem feeding insects, because of the strong negative pressure in the xylem, they have very pronounced musculature in their quote unquote face. And you can see that in this photograph here. So if you get an insect stuck in a sticky trap and you're wondering, oh, is this a xylem feeder or not? A lot of times the first thing that we look for is the face and to see whether they have these protruding or prominent muscles associated with their specialized mouth parts. The most common vectors in California are the glassy wing sharpshooter and the blue-green sharpshooter. So the glassy wing sharpshooter found in more of the central and southern part of the state, and the blue-green sharpshooter, which is our most common vector in the North Coast region. Don't let that fool you though, because there are a suite of other vectors, including green and red-headed sharpshooters, 2 species of Pagaronia sharpshooter that we didn't really know that much about before we started some of our long-term monitoring studies, the willow sharpshooter, and then other sharpshooters that are so uncommon that they really don't even have common names. Spittlebugs are also vectors. They're also xylem feeding insects, and they're also vectors of Xylella. So the meadow spittlebug is fairly common on ornamental plants, and the Afrophora spittlebug from our surveys we find quite commonly in the Mayacamas Mountains in Napa County and also in Sonoma County. Clastoptera is another spittlebug species. It appears to be less common than either the meadow or Afrophora spittlebug, but we have found it in our traps. And just so you know, it's not just because we're out, doing these extensive surveys and looking for these species, but these 2 photographs were submitted to me by growers within the Napa region who had found Afrophora in their vineyards. So we do see the adults. So one was trying to make friends here with this vineyard scout that's on her arm, and then another one that was submitted and it was found on a grape leaf during the growing season. As I mentioned, alternate hosts come into play. These sharpshooters are overwintering as adults. The sharpshooters are, not the spittlebugs. And so they're feeding on alternate hosts during that time. They're riparian hosts, but we also find them in ornamental vegetation, such as in these lush gardens that are surrounding wineries and houses in Napa County. The glassy-winged sharpshooter's most important overwintering habitat are citrus groves, and the blue-green sharpshooter that you can see photographed in the video here are pretty common in riparian areas, and then, as I mentioned, also in ornamental vegetation near structures. Some of the hosts that are important include any non-cultivated or escaped grapevines, as well as our wild grapevine species, Vitis californica. This photograph here was taken of a riparian vine in Napa, and you can see with the leaf scorch here almost looks like, or that it could be Pierce's disease symptoms. Blackberry is another very common host for blue-green sharpshooter. Elderberry, which I feel like receives a little bit less attention, but is no less important as an alternate or overwintering host. Periwinkle or vinca, beautiful purple flowers right near one of our monitoring traps. Mugwort, which is an Artemisia species that we find growing in the riparian areas, and we've collected large numbers of blue-green sharpshooters off of mugwort. And stinging nettle is another very important host. So let's talk a little bit about the annual disease cycle. And this is specifically for blue-green sharpshooter, which is our most common vector on the North Coast. So from November through February, as I mentioned, those sharpshooters are overwintering because they need to continue to be feeding. And okay, sorry. I was just checking the chat to make sure everybody could hear me and everything. Okay, so as I said, they're feeding in the November through February period, doing that mainly outside the vineyard because, as we know, the vines are dormant. And the vines during that period, not too much exciting is happening, except that the cold temperatures that we get over the winter are curing vines, only vines that were infected after June. And I'll talk about that a little bit more as we go through the disease cycle. So in March through May, that's when because in response to the flush of growth in the vineyard, the blue-green sharpshooters are moving into the vineyard, feeding on the vines, and that is the most important time of year for transmission to happen, for vines to become infected with Xylella and then develop Pierce's disease. So in order for a chronic infection to develop, which means that the disease is there to stay and it's going to kill that vine, the transmission has to happen during this period. And the reason is that Xylella, in order to survive the winter, has to move from the shoot or the one-year-old wood into the permanent woody structures of the vine. And that happens when infections and when sharpshooters are feeding on the vines in that March through May period. So that's a really, really important period as those infected sharpshooters move into the vineyard. In July and August, we still find blue-green sharpshooters in the vineyard. That's the summer generation of blue-green sharpshooters. They're feeding on the vines. They may, in fact, transmit Xylella during that time, but it's not really important epidemiologically because they feed at the shoot tip, and there's not generally enough time for Xylella to move from the shoot tip into the permanent woody structures. And if it doesn't move into the permanent woody structures and we get sufficient cold temperatures during the winter, then those vines will recover and they will never really develop a chronic Pierce's disease infection. In August through October. In August, usually mid to late August, is when we start seeing Pierce's disease symptoms become really obvious. That's when, if we're having a bad PD year, my email and my phone start blowing up saying, oh my gosh, there's Pierce's disease symptoms everywhere. Is this PD? And also that's an important point of time, because blue-green sharpshooters are still in the vineyard, and they're leaving the vineyard to overwinter towards in September and October. But before they do that, they're probably picking up Xylella from infected vines in the vineyard. And then they overwinter, and Xylella stays in their mouth parts the entire time when they're overwintering, so that when they move back into the vineyard in March or May, they're able to transmit Xylella back to healthy vines. So it's a really important time, not only for symptoms, but also we think for blue-green sharpshooters acquiring Xylella from infected vines before they go to overwinter. So Pierce's disease is episodic within the North Coast region. There's some kind of low level of disease present somewhat continuously, particularly in areas that are really close to the riparian habitat. But every once in a while, we have outbreaks. And during those outbreaks, we get massive spread of the disease. And some vineyards and vineyard blocks, particularly those that are close to riparian or other suitable habitat, can have populations of 50 or 60% of the vines infected with Xylella. So I've heard of one vineyard that during the outbreak period had 40% of their vines out of production due to PD. So it's there pretty much continuously, particularly in the North Coast region, but we get these outbreaks, and it's therefore a fairly costly disease for the grape industry throughout the state. I'm going to touch just briefly on the typical symptoms of Pierce's disease. Leaf scorch is something that you're probably fairly familiar with. Leaf chlorosis may be a little bit less familiar. So in the spring that the vines become infected, they won't show these chlorosis symptoms. But if they have a chronic infection, or they were infected a year or two previously, in the spring when when the buds are growing and everything is pushing, you get this chlorosis look to the leaves as compared to a healthy leaf. So that's something to look for in the spring. Other symptoms that generally you start seeing in August and September would be the matchstick or stuck petioles, where the leaf blade will fall, but the petiole will remain attached to the shoot. We see uneven lignification, or within the shoot, these islands or green islands that are not lignifying. And then we also see shriveled clusters. As I said, this is a fatal disease at this point to vinifera, and so eventually you start seeing stunting, decline, and vine death. I want to emphasize that in the first year, or in the year that the vines become infected, those symptoms are pretty mild. So that photograph that you're looking at, that vine tested positive for Xylella. It is infected with Xylella, but it was inoculated by our research team. And it doesn't have very many obvious symptoms. The only really obvious symptom is this shrivel of the clusters. And so even a vine may be infected, particularly in that first year, and not have really obvious symptoms. And that's really important for what I'm going to talk about later. Other diseases or disorders can have symptoms that appear similar to Pierce's disease. So for instance, when a vine has ESCA or measles, it's a fungal disease that infects the wood. You can get a leaf scorch. Of course, the pattern is a little bit different than what you see with PD, but you also see some vine decline, and the clusters will shrivel, although they'll also have what gives the characteristic symptom of the measles on the berries. Although once the berries get dried and shriveled, sometimes you can't see those symptoms very well, so the berry shrivel could be confused with PD. Some people also confuse sunburn or heat damage with PD. In that case, sometimes just the exposed clusters or exposed berries will be shriveling, and the pattern of the damage to the leaves or the scorch to the leaves is really different than what you see with PD. Okay, so I'm going to talk now a little bit about PD management and what are the most important steps are for Pierce's disease management. In the North Coast, riparian revegetation has been our single most important thing that we've been able to do for Pierce's disease. And what we're doing when we do riparian revegetation is removing those alternate hosts for blue-green sharpshooter and thereby reducing the habitat that they have for overwintering. And don't forget that blue-green sharpshooter is not our only vector species in the North Coast. So things like the red-headed and green sharpshooter are gonna have main alternate hosts are grasses. And so areas that have a grassy ditch or that may be near a golf course, those can also be alternate hosts and alternate places where a refuge for the red-headed or the green sharpshooter. Of course, there's not much you can do if you have a golf course. You can't really revegetate that with some other species, but it's just something to know that those grasses can also be refuges. With the spittlebug vectors, this is a little bit more complicated. So there are alternate hosts, particularly for Aphrophora, were not well characterized previously. And for meadow spittlebug, it tends to have a very wide alternate host range. So dealing with the alternate host for meadow and Aphrophora spittlebugs is a little bit more complicated. From our studies, we know that bristly oxtongue, curly dock, poison hemlock, and short pod mustard were very important host plants. And I want to point out that these are the host plants for the nymphs. So what you're seeing here, this spittle is produced by the spittlebug nymphs. And so if you start, and it's there to protect them, and they're feeding inside of that spittle. So if you start, kind of brushing that spittle a little bit, here are the nymphs feeding within that host. And they tend to be, it's pretty obvious once you train your eye for it to find these spittles, particularly when the nymphs are larger. But they do tend to be at the base of these host plants. And so trying to reduce them as a host can be a little bit more challenging, particularly when you're working in a hillside vineyard and you need to maintain by regulation some sort of ground cover. So that's the biggest challenge that we have with the Aphrophora spittlebug host management. What we did find is that cultivation was a good way of reducing those meadow spittlebug hosts. But again, that's cultivation mowing, unless it's a very, very, very low mowing or like almost a scraping of those hosts. It doesn't reduce the amount of plant material that's available enough because the spittlebugs are feeding so low on those alternate hosts. And the other important management strategy that I can't emphasize enough is the timely removal of diseased vines for the vineyard. And so for anyone who was around working as a PCA or vineyard manager in the 90s during the last PD outbreak, it was always sort of common knowledge that it didn't matter. Those PD vines were going to die anyway, so you didn't really have to proactively remove them. But once they were dead, sure, you were going to remove them and replant them. Meanwhile, during that entire process of when they're dying, which could be 2, 3, 4, 5 years, they are a reservoir of inoculum, of Xylella that the vectors can feed on, pick up, and then perpetuate the disease. So we do it's a little bit of a change in philosophy now, but we do think that those vines need to be removed from the vineyard as soon as you become certain that they do in fact have Pierce's disease. And you can see in this photo that what they tried to do was say, okay, well, treat it like a trunk disease and say, well, the Xylella is not totally throughout the vine. So what we're going to do is we're just going to lop off the trunk. We're going to retrain a trunk sucker, and then we'll have a healthy vine. Well, it turns out that Xylella was throughout the entire vine, and that's not a strategy that works. So severe pruning and retraining of diseased vines, while it may be important for trunk diseases, it's not something that's recommended for Xylella. As you can see in this manipulated study, it might appear to work in the first year because the vines appear to recover, they appear to be growing normally, but then by the time the second year comes around, they are back again showing symptoms, and they will never recover from that. So the best strategy is just to get them out of the vineyard when you can. Here's just another example of that. You can see in the background the normally growing vines, but in the foreground, these are the vines that are still affected with Pierce's disease and will not recover from that. A similar strategy here where they tried to lop off the cordons and retrain them, and those vines are still stunted. So I think a key aspect of that is to make sure that your workforce in the vineyard is very well aware of that. So my 5 years of mapping thousands of vines, what I've noticed is that our workers are very conscientious. And so when they see an arm, that doesn't appear to be growing well, a part of the cordon, when they see a vine that's not growing well, they'll just lop it off and try to retrain it, try to fill in that space. But if that actually is PD, what we need to do is communicate with them that, no, we don't need to try to recover those vines. What we need to try to do is mark those vines and get them out of the vineyard. And so I think it's really important, particularly in the areas where you know that you have PD, to make sure that your workforce that is in the vineyard is aware of that and can take the appropriate actions. So a robust training program on PD symptoms and management for our workforce would help a lot with us being able to timely remove these disease vines. Okay, so those are the two most important strategies, but I want to talk about a few other things that come up questions-wise with regards to management. So insecticides, it's, of course, we know that we have a vector, so one of our knee-jerk reactions is like, okay, well, if we had fewer insects, maybe we wouldn't have as much disease. Well, that's worked pretty well for us with Glassy wing Sharpshooter. It's allowed us to contain the Glassy wing Sharpshooter in the southern part of the state, and it hasn't moved very much and established in the north or the coastal areas of the state. Although there are other things that are being done, such as the inspections of plant material moving out of those areas that are also helping with that. And we have some concerns that have come up more recently around insecticide resistance of Glassy wing Sharpshooter, showing some resistance or tolerance to some of the insecticides that have been used long-term in those programs. So it is a fairly efficient for Glassy wing. For blue-green Sharpshooter, insecticide management has never been all that shown to be that effective. That's two reasons, I think. One is that it's a very efficient vector, and secondly, in that it's a native insect, and so the populations are already quite low, and the timing of when we're trying to use those insecticides has been in the spring. And so we've tried to control the adult sharpshooters in the spring as they're migrating into the vineyard. And I think that's a pretty difficult proposal. It doesn't work very well with systemic insecticides because the vine is not taking up enough of the active ingredient to kill the blue-green sharpshooters at that time of year. And with our contact products, I think it can be just difficult to make contact with the blue-green sharpshooter and therefore to reduce their population. So we do have some questions, however, though, about controlling or potential use of insecticides during the summer generation to control those insects, because if we were able to reduce the number of insects that are infected with Xylella that are leaving the vineyard in the fall to overwinter, there's a possibility that that might be helpful as one portion of a disease management program, but that still has to be tested. Biological control, as I said, blue-green sharpshooter is a native insect. It generally has pretty low populations. So in March to October of 2020, we had 192 traps throughout Napa County, and we only captured 186 blue-green sharpshooters. So we know that the populations are already generally pretty low, and there's probably already a fairly good biological control. So any augmentation so far has not been found to be useful. Insectivorous birds, there was a lot of attention a few years ago around western bluebirds as potential biological control. I'd say insectivorous birds are a good thing. It's nice to be out in the vineyard and have good bird wildlife. And so it can't hurt to have bird boxes, although bluebirds are gonna forage on a variety of different insect species. And so, yes, there may be some control of the blue-green sharpshooter, but probably not exclusively. Barrier treatments are another option. They haven't been very well studied for blue-green sharpshooter, but in some studies with glassy wing sharpshooter, it showed that the surround barrier both reduced feeding and egg laying. So not only do the sharpshooters not like to feed on these vines that have surround, but also they're not laying eggs in them quite as much. And so although it hasn't been widely tested, people, particularly in areas where there's a lot of Pierce disease pressure, have been using surround as a potential barrier. Of course, you have to wonder if you use surround in a certain area, if that's just going to drive the sharpshooters into another area of the vineyard, and how much area would actually have to be treated with surround in order to to be effective. I think the other problem is that the vines are growing so rapidly during the spring. And so being through the field and being able to keep the new leaves or the new foliage protected with surround is another challenge. As many of you have followed Dr. Walker's career and his breeding efforts, we now have PD tolerant cultivars with a gene crossed and back crossed into vinifera, so that we these tolerant cultivars are about 97% vinifera, and they're similar to Chardonnay, Zinfandel, Petit Syrah, and Cab. So they're pretty popular in the southeastern part of the U.S. where vinifera really can't be grown because of the PD pressure with glassy wing sharpshooter. And there are people that I know of in the North Coast region who are also planting these vines. It's important to note that they're tolerant, they're not fully resistant. So that means that they will host a very low concentration of Xylella within them. We don't really understand if that would be enough to create problems for if insects were picking that up, but in any case, it's a really great option. And then Dr. Lindau's work with a naturally occurring beneficial bacterium has a potential to both pre and post-infection cure vines that have PD. And so it works by triggering a plant immune response and then being able to eliminate Xylella from the plant. And so that is still under investigation in the field, but there is some potential for that I think we can look forward to. I think one of the remaining questions that we need to answer is what triggers an outbreak? So why did it take 20 years for us from the late 90s to the mid 2013 to have an outbreak? And we don't really understand that because we don't have a lot of long-term monitoring data, either of the vector blue-green sharpshooter or the disease in the North Coast region to be able to understand that. So this year, with the support of the Napa County Wine Grape Pest and Disease Control District, we initiated a vector monitoring program that we intend to carry out over the long term in Napa County to give us more information about that. And what we've done is identify over 190 different sites. Some of those are riparian traps. Those are the blue ones. Some of those traps are building traps. That is, they're within 50 meters of a building, which would be a winery or a house with a garden. And then the other are between sort of the riparian and the building zones. These data are available live at this website. We have, just like Johns Hopkins has done for us for COVID, we have a blue-green sharpshooter dashboard. So I'm sure it's not going to get the millions of hits worldwide that the COVID dashboard does, but within our region, I hope that you'll look at our blue-green sharpshooter dashboard because you can get long-term data on where and when we're catching blue-green sharpshooters. So you can see that in 2020, we had a peak in April and May, which would be expected because that's when the sharpshooters are moving from overwintering habitat into the vineyard. And then in August and September, we had another small peak, which is the summer generation that's both moving within the vineyard and then moving out of the vineyard towards the overwintering habitat. We have the traps categorized not only by trap location, but also by region. So you can see within the regions of Napa where we're collecting the most blue-green sharpshooters. Coombsville was a definite hotspot. Mount Veeder was a bit of a surprise. I didn't expect that many blue-green sharpshooters on Mount Veeder, but we found quite a few there. Gordon Valley, Rutherford, and Oak Knoll were also pretty populous areas. And I would like to thank everyone for their attention and to everyone who's been involved in this project, as well as the growers who have given us access to the field sites. And as I mentioned, the PD GWIS board that's provided funding for much of this work, as well as the Napa County Wine Grape Pest and Disease Control District. And I spoke pretty quickly, so it looks like we got some time for those questions.
Thank you, Monica. I am going to have Aubrey take over to feed the questions to you that we have in our Q&A.
Alrighty, thank you, Monica. The first question we have here is how are all of the increasing numbers of environmental regulations affecting PD strategies?
As far as what you can and cannot do with regards to host removal, I guess. So that's one of the things. It's great that we have a lot of these strategies that are, protecting our watersheds, reducing runoff from the vineyards. But as you could see from my presentation, that does lead to the, presence of hosts and host plants that tend to not be managed and can be problematic. So I think we could, from Cindy's work with Tika and red blotch, we see that. And then, so we also see that with PD. So it's a good thing, but I think what we need to do is be a little bit more maybe conscientious about what types of species we're we're propagating in those areas, and that they're not problematic for some sort of pest and disease management that we're trying to do in the vineyard.
Alrighty, the next question is, after roguing diseased vines, is fallowing recommended before replanting? And is PD vectored from residual plant material left in the soil after removal of diseased vines?
Okay, yeah, so it wouldn't be vectored from residual plant material. So if you're a xylem feeder, you're not going to be feeding on residual root material. So you need xylem feeders prefer to be growing in sort of actively growing regions of the vine. So they're like blue-green sharpshooters feeding at the shoot tip. They're feeding in this young tissue that's much easier to feed in. And so I wouldn't expect any movement from residual plant material into new vines. They're not feeding on that and becoming infected. And is fallowing recommended before replanting? Not necessarily, except that when we're in an outbreak, you're removing the vines that have symptoms. But as I showed from the inoculations, there's going to be some amount of vines that are still infected but don't have symptoms. And so it may, if you're in like the peak and you've seen a lot of movement of PD, you might actually want to consider just making sure that you've gone through 2 years of roguing diseased vines before you start replanting. And that's just to kind of make sure that you've gotten all of those host plants out of there. But if we're not in an outbreak period, if it's just like, oh, this vine is here and it's probably had PD for 5 years and there's not a lot of other, incidence levels are really low, less than 1%. I don't think that you need to fallow. I think you could just go ahead and replant. And it's not that you're going to get PD by any kind of root grafting or anything like that.
Okay, awesome. The next question is, have you studied nighttime spraying versus daytime spraying? And are there any differences in those timings regarding efficacy?
Yeah, that's a good question. It's something I've thought about because it's not really well understood where the blue-green sharpshooters are at different times of the day or night. I haven't studied it, but there may in fact, any insect is going to have temperatures that it's most active at. And so it may be that during the day, the temperatures are warm enough for the insects to be active, but at night, they're really not as active. And it's just going to depend where they are when they're not as active. Are they still on the vines when they're not that active? Or are they have they moved back into the riparian areas? Have they fallen down into the cover crop or some other vegetation? So it would be a good thing to investigate, I think. But unfortunately, we can never find enough blue-green sharpshooters to really understand it. So,
Awesome.
yeah.
Okay, the next question is, has anyone tried the yellow sticky tape like we used years ago for leaf hoppers?
People have tried barriers. Somebody has put up some of that yellow sticky tape. If anybody's ever tried to use that stuff, it's horrible. So, and mostly in my experience, what you're catching is not, sharpshooters. You're catching like flies and other insects. So, it's not, in the priority list of things to do. It's probably not something I would prioritize.
Right.
Making sure that you're getting rid of the alternate hosts and getting rid of your disease vines, I think is going to be, better bang for your buck.
Mm-hmm. Alrighty. The next question is, what are your thoughts on the bacteriophage injections for Xylella control?
Yeah, that's a good question. I realize that's getting a lot of press right now. I haven't seen any convincing data either from the greenhouse or from the field that suggests that is going to be an option that will be useful.
Okay. The next question is, do you feel that vine mealybug treatments in the spring will help lower sharpshooter populations in the vineyard?
Yeah, that's possible. Because, and so what it's not gonna do is reduce the incoming sharpshooter population. So those those populations are still gonna migrate in because they're doing that in March and April, and your vine mealybug treatments are usually not going on until May. But say you're using a systemic treatment for vine mealybug in that late spring period, there's a potential that would then reduce your sharpshooter populations, your summer sharpshooter populations, which would then potentially reduce the number of infected sharpshooters that are leaving the vineyard to overwinter. So yes, but it wouldn't reduce transmission in that treatment year.
Okay, got it. The next question is, this person has found spittlebug nymphs inside cartons protecting her 1-year-old vines this spring. Any ideas on how to minimize this?
Oh, yeah. Yeah, that's a good question. We didn't finish it, but we did do some work with backpack sprayer of insecticides on our alternate hosts. And so I think that's probably something that it would be helpful, would be some sort of insecticide that you are using, that you're treating with a backpack inside the carton to kill the nymphs. Of course, you're probably going to have more success doing that when they're young nymphs because they'll have less spittle than when they're older nymphs. But the other things that we tried to reduce the spittle populations on the alternate host was flaming. So obviously you're not going to want to flame your 1-year-old vines, but that worked pretty well. And we tried surround, but it didn't really help. So I think you would probably be limited to some sort of insecticide.
The next question is, whatever happened to the Sandy Purcell work on changing the vegetation near the riparian zones? And are any of those sites still being monitored?
Okay, thanks, Kent. So I don't think any of Sandy's specific sites were being monitored, but what his work did was kind of trigger that big revegetation effort in Rutherford. And what we know from long-term, both grower monitoring and our own monitoring efforts, is that revegetation work where they did a massive swath of the Napa River was revegetated through Rutherford, and they're now extending that through Oakville and Oak Knoll. But the blue-green sharpshooter populations in that area are much, much lower than other parts of the Napa River where it hasn't been revegetated. So yeah, in not Sandy sites directly, but yes, we do know that revegetation on a large scale does reduce both the blue-green sharpshooter numbers and then also the associated PD, because both just south of that and just north of that, we still have a lot of PD, and then we don't have as much PD in those sites that are along the Rutherford branch of the Napa River.
Okay. I think you went over this, but maybe if you could just remind us, are there any updates on less susceptible varieties to consider in riparian areas with a history of Pierce's disease?
Yeah, so it's a continuum. Of course, there's the Andes varieties that have tolerance, but of the vinifera, classic vinifera varieties, I do know that Chardonnay seems to succumb the worst. And so we had a site where we inoculated both Chardonnay and Merlot, and the Merlot seemed to do a little bit better than the Chardonnay, didn't collapse quite as much. So there probably is a little bit of a continuum, but when you have such high pressure, there's not going to be anything that's really resistant, and you can still, I mean, even some cab blocks experience a lot of loss when we're having an outbreak. So yes and no, I guess.
Okay. What time of year are the spittlebugs most active in the vineyard? And is it similar to blue-green sharpshooter? And where are they most likely to feed on the vine?
Okay, so in the spring is when we are seeing the Spittlebug Nymphs on alternate host. So we start seeing them probably sometime in February, and we see Spittlebug Nymphs through May. In May, those nymphs become adults, and so we start seeing adult Spittlebug Aphrophora in the vineyard starting in May, and they'll feed. They only have one generation a year, and so those adults are going to feed in the vineyard and on other hosts, kind of like from mid-May onward. We don't really know where they're most likely to feed on the vine. It's possible. They're a much larger insect, and they're a little bit closer in size to glassy wing sharpshooters than they are to blue-green sharpshooters. So what we're worried about is that even like blue-green sharpshooters feeding at the shoot tip, we're worried that they're actually feeding lower on the vine. And then that would be a problem for transmission, because if they're feeding in June and they're feeding lower on the vine, Xylella would be inoculated lower, and it could still move into the permanent wood. So that's a good question.
Okay, this next person is wondering of the efficacy of Admire applied via drip on Sharpshooter.
Okay, yeah. So as I said, in the spring, efficacy is really, really low. So if you're trying to control the population that's moving into the vineyard from the alternate or the overwintering areas, Admire through the drip in the spring is not a good choice, because it's not that it's not effective, it's just that it's not taken up by the plant in high enough concentrations to kill the sharpshooters. So when it may be effective would be on the summer generation. So that would be the summer generation we start seeing in June and July. And it's possible that if we had a sufficient amount of Admire in the vine at that time, that we might be able to reduce the summer population, but we have not studied that.
Okay. The next question is, what threshold constitutes a Pierce's disease outbreak?
Yeah, that's a good question. I don't know about thresholds. I'd say generally we have really low incidence, like maybe less than 1%. When we have outbreaks, our average, so we monitored 30 different vineyards, and I think our average was about 10% PD throughout those vineyard blocks as an aggregate. As I said, some of them will have like 60% disease incidence, but it has to do not only with how much incidence you have in your own vineyard, but then also how much there is at how many different sites. So.
Okay, and finally, there are a couple questions on fire damage and how that has affected blue-green sharpshooter. And one person asks, is this a good way to replant all the non-host vegetation and to have trials with newer possible non-host plants?
Yeah, so I guess that would be one silver lining of some of the fires, is that it does give you a chance to get in there and try to revegetate those areas. The one thing I've learned from the fires is that there's very few plants that are killed outright through the fires, that most of them, the root system will survive and you start seeing them grow back. So the fires alone are probably not going to be enough to like get rid of your blackberry, right? You're still going to have to go in there and either dig up the roots or have some kind of grazing in those areas to permanently reduce things like blackberry and vinca. And then some of the plants like the mugwort is seasonal anyway, and so it probably also wouldn't be killed. But yeah, it does give you a really kind of clean slate to at least be able to get in and start a lot of that work. And I would encourage people to try and do that and get permitting in order to do that.
Alrighty, and then there's another question on Admire. If
Mhm.
applying foliarly will be any more effective in the spring?
Yeah, more than through the drip, but again, we've never really seen great impacts of those foliar applications. So it's yes, and then also it might reduce populations, but how long would the insects be feeding before they would be killed? And so transmission may still happen. So none of those foliar applications have been, all that effective either. So.
Alrighty, last call for questions to Dr. Monica Cooper. I think
That's good.
it.
Thanks, Aubrey.
Thank
Yes.
you very much.
Thank you, Monica, for presenting today.